Welcome to Translating Proteomics, where we explore the science of proteomics and its growing impact on basic biological research, drug development, and more. In this episode, we cover biomarkers and specifically discuss why new biomarkers are difficult to find, why most biomarkers are proteins, and how we can improve the biomarker discovery process. And now your hosts, Parag, Malik, and Andreas Humer of Nautilus Biotechnology.
Welcome back, everyone. Today we're going to talk about a topic that is central to so much of biomedicine, biomarkers. Diving in, first question, what is a biomarker? I've always thought of biomarkers as something that you measure, a protein, a metabolite, that tells you about the state of a disease, whether one has it or not, if a person's going to respond to a drug, if a person's currently responding to a drug. What is a biomarker to you?
And any type of endpoint or answer you want to have from a biological system is probably encapsulated by a particular marker, which I would call a biomarker.
If one looks throughout the literature, there are literally tens of thousands, hundreds of thousands of papers maybe that have been looking at biomarkers or claiming to have found a new biomarker. If one looks in the clinic, there are maybe 100 or so FDA-approved biomarkers. That seems like a terrible disparity, which speaks to it being really hard to find biomarkers. Why is it so hard to find biomarkers?
I guess there's two camps. The one camp says, they're just very rare. They're, really rare events that ultimately create these biomarkers, and we have to have super sensitive, tools to sort of find them. There's another camp that says that maybe the way we look for biomarkers is incorrect, and that makes it very hard for us to find them, because we look at the wrong time scale, or we look at the wrong places.
Yes.
Typical example there is a lot of people start biomarker discovery in blood, which is the most complex mixture that we have available, in humans. And it may be smarter to start studying and finding a biomarker in the particular tissue of interest, and then see whether we can replicate that later in blood.
So I'm going to put you on the spot. You mentioned there's these two camps. Which are you in?
I am actually in the camp that we're, approaching this mostly, in the wrong way. I don't think necessarily the biomarkers are rare, super rare molecules. I think they're typically just hard to find because we're trying to find them in the wrong way.
Okay. All right. So I'll just add on that topic of we're looking at it in the wrong way. I'm going to say that I do think they're hard to find. I think they're hard to find for a couple of reasons, and some of those are technological. One, when we look at just human variability, I do think there's a tremendous amount of human variability over time, what we eat, how we interact in the world. And so getting beyond that variation to what is caused by the disease, there's a huge amount of noise that you're looking for low signal in. Number two is just our methods for collecting samples. If we leave a tissue, a surgeon takes something out, a tumor, leaves it on a gauze pad for a minute, or 5 minutes, or 10 minutes, or hours during a surgery, lots of biology is happening during that time, which is obscuring the signal of that tissue. So and likewise, from a tube of blood, we know that there are metabolites that are gone within seconds of taking blood out of the body.
Correct.
So these pre-analytical variables are tremendous. And then on the technical side, I agree with you that I think the technical challenge of measuring very rare things in very complex samples is an incredibly hard problem. And so you layer those on top of each other, and I do just think it's also just very hard to do.
But I think that could be overcome. If you think of other problems in physics, where, we're trying to capture the presence of a neutrino, right?
Sure.
We just do massive amounts of measurements. We observe for a very long time.
We did spend billions of dollars, trillions of dollars in order to make that detector.
But we tried in a massive way. And I think we just simply don't have the data points yet, and we need to create them. We need to create them with better tools where we just take many, many more data points. And then I think it will be much easier to find even those rare events. So, but I think when I think to many conversations I had with other scientists, I often see the biomarker concept being reduced to the smoking gun. There's something there that otherwise isn't there. And only if we find that piece can we explain, for example, a disease or measure a disease. And I think that general is probably not the definition of a biomarker. Mm-hmm. It's much broader. It is essentially, an endpoint or a particular change that you're trying to capture.
Yeah, I guess the way that we use them is we measure something, whatever that something might be, and we use it to say, all right, well, you have this disease, you might have this disease, you might respond to this drug, you are responding to this drug. And so functionally, they are signals that we look at to help us make decisions.
Yeah, correct. And I think a lot of people do not recognize that within, for example, the drug development process, there's many biomarkers that are being used. They're never being published, or they're not being talked about as the biomarkers we typically talk about, which diagnostic biomarkers. But there's many biomarkers we actually already use in the context of, for example, drug discovery.
So you mentioned that there are lots of different types of biomarkers. And in your mind, what makes a good biomarker?
That's a very good question. I guess, again, it's a little bit related to what you're trying to measure. But I think a biomarker should be, obviously consistent. It should be something that you can measure relatively easily. So, if you have a biomarker that is in your brain, but you want to measure it in someone's lifetime, then it's a bad biomarker because you can only find it when you're dead, or you can measure it when you're dead. So I think that depends a little bit on the particular biomarker, but certainly it has to be in a market that can be easily accessed and reproducibly measured.
Okay, that makes sense. I mean, coming back to proteins, for the vast majority of biomarkers in use today are proteins. Why?
I think they capture the right time frames, right? When we think about, how changes are happening in biological systems, there's biomarkers, in the metabolomic space, right? Where, something that you ate immediately has an effect on your body, and you can sort of notice that immediately, right? Mm-hmm. But typically, when we think about diseases that happen over long periods of time, proteins are the ones that capture those changes, right?
So you don't think it's because proteins are actually the drivers. You think they're just they happen to live at the right time scale.
I think they're living at the right time scale, yes. I think that's why proteins are biomarkers. I think you also have to look at the half-life of things, right? I mean, sugar has a very short half-life in your body. A protein typically stays for weeks. Mm-hmm. And so they're much easier to capture as a biomarker than a small molecule, for example.
That's interesting. I totally disagree with you.
Oh, perfect.
I agree with you on time. I think that time is really critical, and we'll come back to talk about time more. But I think that proteins are actually doing the work throughout our body. And so if you had the option to look at the element that was doing the work, that was part of the mechanism, versus something that was secondary or just a correlate, you would prefer to look at the thing that's doing the work.
Certainly. Yeah, absolutely. So that brings back the conversation on, what I call the active form of the protein. Mm-hmm. That would be the part that you have to capture. So you can actually say, yeah, this particular mechanism or this particular transition just took place, and I captured that, and that's the biomarker for that particular transition.
One of the things that I think about a lot is time when it comes to biomarkers. And when you think about most biomarkers in use today, they are single time point biomarkers. You measure something and you say, oh, okay, this is what's going on. I'd love to hear your perspectives on this. Do they have to be? Is that the right thing to do?
Well, I think you just gave yourself the answer why it is incredibly hard to find biomarkers, because if you just keep looking, using cross-sectional studies at 8 random time point across a population, it is incredibly hard to find a biomarker. And I think the better way to approach this is really, putting measuring stick that you have, your tools that you have available, and probe the right time points, right?
But when a patient walks into the hospital, you don't have any control over where they are in their process. They're there because they don't feel well.
That's correct, but I think that's where we may have to make changes in the future, or we have to have more specific measurements that are, understood across time frames. So, for example, if a patient shows up and he's non-critical, we have a biomarker for that. And then if a patient shows up and he's critical, then we have a biomarker for that. And there may be 2 different biomarkers, right?
Yeah. I guess the way that I think about it is a little bit like that game show, Name That Tune. And in Name That Tune, if I played you one note on a synthesizer and asked you to name the song, it would be virtually impossible. Not totally impossible, but virtually impossible.
Virtually impossible for me, for sure.
and that's kind of what we're asking of these biomarker tests. That's the bar that we're saying, yeah, we just need to measure you once with no extra context, no proteoform information, no texture to know it's an electric guitar or a violin, and figure out what's going on. On the other hand, if we gave you a chord, if we gave you the type of sound, is it a violin or a guitar? Is there distortion on it? If we get a whole measure, it becomes so much easier to guess the song and accordingly where the song is going to go. So to my mind, we absolutely need to get into a world where not only are we measuring, one thing at one time abstractly, but we're measuring chords across time.
And I think that has been, the graveyard of many measurements in the biomarker discovery space, that people just keep looking at diseased, non-diseased at a random time point. I think people are sort of starting to go beyond that. I would again say the tools we have are probably not sufficient yet, because I think, as you mentioned, we probably want to look at proteoforms, because they usually are almost like a timestamp, within coming from the cells. I think we need massive amounts of data, and the way I envision this is simply, we can probably calibrate, certain diseases and how they progress over time, through multiple measurements across multiple patients, right? And I think they eventually will find a way to line them up. And once you have that map, you can basically, measure a patient once or twice and then say, yeah, by the way, you're on this progression, you're on this map, right? And this temporal map, and this is what most likely can happen next, right?
Yeah, so that you have the entire sheet music.
Yes, exactly.
And you're trying to land someone within that sheet music.
Exactly, yeah. So what do you think are the next steps people really have to think about if you want to break beyond this pattern of, these, simplistic studies that we're currently doing?
So one is, I think we have to, one is a big ask, just changing the way that we think about biomarker studies and the way that we sample people. So when we now have things like these smartwatches where we're getting continuous measurements of people. So it's not just a single time point. We have tests like Cologuard, which are done in your home, which makes the sampling problem easier. So if we can shift the paradigm from one that's constantly reactive to one that's proactive, we have a baseline for each person. We get routine sampling of them. This is why some of the data sets from the Veterans Administration are so powerful, because the soldiers there, the members, are sampled at a routine interval at a minimum every 6 months.
So yeah, then the question comes up, why would, I understand that, for example, mice are not humans, but why wouldn't we do that on a massive scale on another organism like a mouse and first get the tools worked out before we then trying to apply it to humans?
Sure. I think you answered your own question. Mice are not human. The amount of variation that mice experience is much lower than your average human. But in terms of building up the capability, I think that's a very reasonable capability. That's a really reasonable thing to do, to build the tooling to be able to do that. So that's number one. I think we need to shift the paradigm towards a more multi-sampling, sample even when you're healthy, not just when you're sick paradigm.
Yes.
Number two is I think we need to think about that different biomarkers may have different properties. So, for instance, when we think about the kinetics of different types of biomarkers, there may be things that we want. So for diagnostic purposes, saying that somebody has cancer, for instance, we want a biomarker that has a very long persistence time in your body so that as the tumor grows, the biomarker builds up and builds up and builds up and builds up. On the other hand, if we're looking for a biomarker of therapeutic response, are you actually responding to this drug that I'm giving you right now? We actually don't want that. We want a biomarker that has very fast kinetics so that I treat someone, I see a response, I measure it at a defined window, and then it goes away so I can then come in with another drug and see a different response. So I do think that they're just changing the narrative around biomarkers and what are the characteristics of a good biomarker. I think the third aspect we really need is we don't know much about the basic biology of biomarkers. We've done tens of thousands of studies looking at healthy people and sick people or responders and non-responders. But if you ask most people, how did that protein get into blood? How big did the tumor have to be in order to see how much of that protein in the blood? These very fundamental questions of biomarker biology. I think we need to put in more work into that. Where did biomarkers come from? How did they get there? How did they behave? And answer those questions.
Yeah, you just mentioned, this probably very simple potential approach that everybody could do before actually doing a measurement, right? I mean, think about the concentration you would have to pick up of a marker in the blood, for example, right? You can easily do that. It's not even difficult math. Yeah, I think that would be probably a good idea to, as part of maybe a proposal, like if you want to measure that marker, how sensitive do you have to be to actually pick it up, right? Given that we know how much blood is in humans and how many cells we have and how diluted it will be. Yeah, absolutely, I agree. So again, I think that all points to, more measurements.
Yes.
Protein measurements, as we already said, right? I think this points to the proteoforms, because I think they have these unique capability of capturing time points within biological systems.
Yeah.
I think that ultimately also points to better experimental design.
Mm-hmm.
Right? We have failed there massively, I guess. What else?
So, I think that's a really good synopsis. I think the last piece that we haven't talked about in the space is really, I guess there are two. So one is the data warehousing. So when we do a biomarker study, are we depositing it in a shared resource where other people can leverage it? And a second piece of that is when we are finding things that are interesting, is there a registry that we can go to? And we'll often find that the people who study cancer, the people who study heart disease. And so, but for something to be a truly specific cancer biomarker, it probably shouldn't change in heart disease. And so we need to be able to take information across domains and be aware of those things. So I think that's another area.
Yeah.
Okay. So if you had to pick one thing, you could solve one problem, which would it be?
I would definitely build better tools.
Okay. So, all right, fine. That was too general an answer. If you could make those tools faster, cheaper, more sensitive, of all the dimensions that you could improve, what's the one dimension that you think is going to be the most impactful?
My vote gets solidly on proteoforms. I'm absolutely convinced that capturing the various active forms of proteins ultimately will be, the the place where you find really relevant biomarkers. So being able to capture those relatively low levels, but very specific forms of them, I think is going to be the key to finding more biomarkers.
Okay. All right. I'm gonna place my money on sensitivity per unit time. Which is totally cheating, and I know that. However, I think that the ability to do studies, very fine temporal sampling, large sample numbers, helps you with that signal-to-noise problem quite a bit.
Yeah, but I think you're going to have to also live with a huge data analysis problem, and also
I am
with
totally okay with that.
I can imagine why.
Shockingly. Thanks for joining us today. We've had an incredibly vibrant conversation about biomarkers and really chatting about both how most of biomarkers today are currently expected to be simple, things that are measured at one point and predictive of everything that might come before. We've talked about how there's a need for extending those towards having more time available, looking at a wider range of things and looking at more detail, as well as to understand the biological context of where these biomarkers come from and the fundamental principles of what makes a good biomarker. The last thing that we really talked about in detail was how there continues to be a need for novel technologies to allow us to dig deeper and faster and more sensitively and to measure other aspects like proteoforms that are hard to measure today. Thank you again for joining us. Look forward to seeing you next time.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.